Welcome to Biology Made Easy. Today we are discussing osmosis in animal and plant cells. Welcome. Learning objectives. Well, this lesson you would need to understand the terms related to osmosis like hypotonic, hypertonic, isotonic solutions, water potential, osmotic potential, and the osmosis process itself, which we discussed in our last lesson. Well, let us begin by looking at osmosis in animal cells. And here we are looking at blood cells. So we'll look at cells. This is how they look like in isotonic solution, in hypotonic solution, and in hypertonic solution. The cells look like this. They are crenated, lysis, they burst in hypotonic solution, and they are good, normal in isotonic solution. Well, how do you prepare these blood cells slides? Because you can't look at them when you pour them into a petri dish or in a container. They should be on microscope slides. So this is it. Blood is obtained in a syringe like this, and a drop of blood is put on a microscope slide like this. And then the solution that is going to be put on the blood here is put on, and the rod is used to smear the blood on to the microscope slide. And then a cover slip is dropped on it. Extra solution is absorbed with a filter paper. So you have the slide prepared, and this microscope slide with the specimen on it can then be observed in the microscope. We discuss how this is done in our lesson on microscopes, which you should please check. So this is what happens with the solutions. Blood in isotonic solution is like this. Blood in hypotonic solution is like this. And blood in hypertonic solution is like this. And you can look at them under the microscope and see the shape of the blood cells as these. Now we want to explain the process that brought about these. And this, we have an illustration here. This is a blood in a hypotonic solution on a blood slide. So the hypotonic solution, the water potential is higher than the contents of the blood cell. So we have a key here. Water molecules move from a region of high water potential to a region of low water potential across a partially permeable membrane, and that's osmosis. So in this one, water molecules moves from this region with a high water potential, that's the hypotonic solution, into the cell contents which has a lower water potential. And the water molecules keep moving in, and the cell swells, and finally the cell bursts, lysis. That is haemolysis. So this is what produced this situation where the cells burst. Now we go to look at this situation where the blood cell is placed in a hypertonic solution. So water molecules move from this region of high water potential to this region of low water potential across the partially permeable membrane, and that's osmosis. So the water molecules keep moving out of the cell, and the cytoplasm shrinks. That is, plasmolysis occurs. When the cytoplasm is shrinking, the cell will be wrinkled, and the cells that have wrinkled are called crenated cells. And this is what happened, and we had this kind of situation. Well, so we consider that animal cells in a hypotonic solution will burst. Animal cells in a hypertonic solution will become crenated and die in both cases. So animal cells are suitable to be in only isotonic solution. And you know, isotonic solution is a solution that will have the same water potential as the contents of the cell, as also the tissue fluid. You see, animal cells are surrounded with tissue fluid. You have capillaries, you have lymph capillaries, and these are the cells. And fluid that surrounds the cells is called tissue fluid. Now, any fluid that is put in an animal cell should be isotonic, should be the same concentration as tissue fluid. Otherwise, these cells will either burst or become crenated. So this is taking note of when infusions, vaccines, serums are prepared. These vaccines and infusions, the concentration of salts should be the same osmotic potential or the same water potential as tissue fluid. Otherwise, there will be problems. Now, the question is, what happens with lower organisms that live in water? How do they control the concentration of substances inside their cells? So we have here, example we use is amoeba. This amoeba lives in water, and the concentration of its cell cytoplasm should always be the same. So when water moves in from the external environment into the cell, excess water gets into a contractile vacuole. The contractile vacuole is a vacuole that forms very tiny, and as water builds in, it grows bigger and bigger and bigger, and then it will move to the surface of the cell and burst to discharge the contents into the environment. And so the contractile vacuole, a new one will be formed, and the contractile vacuoles make sure that the concentration of salts inside the amoeba or unicellular organisms are always the same, constant. And so all these unicellular organisms have contractile vacuoles to regulate the osmotic potentials of their cytoplasm. Very neat. Now, what is happening, therefore, in plant cells? Well, in plant cells, we want to use Rheodis color, whose epidermal cells are colored, so we can see what is happening to the cells when it's placed in different concentrations of solutions. Now, these are epidermal cells. These cells are turgid. These cells, they are plasmolysed. So, how do you get these plant cells to look at? Because you can't see the plant cells as it is in this one. You can't see them. So, we need to get plant cells put under the microscope so that we can observe what is happening inside the cells. So, we want to explain how to get transverse section of leaves. So we have this plant, we take a leaf, we want to get a section, transverse section, so that we can look inside and see the cells. So this leaf is taken and a piece of it is broken with a forceps. And this piece we have here, we get a polystyrene, make a cut through it and push the piece of leaf into it. We want to get very thin sections that we'll put under the microscope to see the cells. So we are going to cut thin pieces from this. So with a razor blade, we slice off as many pieces as possible. As we slice off, we drop into the water in this dish. And a lot of pieces are cut. We look through and pick the tiniest with a brush and put it on the microscope slide and drop the cover slip on it. And this is ready for us to look at the plant cells in transverse section, the leaf in transverse section. This is how they look. This is the epidermis, and these are the mesophyll, palisade, spongy, vein. This is the transverse section, and you can only see them through the process we've gone through. But in this lesson, we want to look at the epidermal cells alone. So how do we get epidermal cells to look at the turgid or plasmolysed cells? This is the process that is taken through. Now, this is the plant. This is a leaf. We can just break one leaf, and we want the epidermis. So we are going to strip the epidermis of this. So with our forceps, we take a piece and we strip the epidermis with a forceps. All right, take it just a strip. And as we strip, we put them in water here. Then we have plenty of strips. We look through and pick the tiniest with a brush and drop it on the microscope slide and drop the cover slip on. These processes, how they can be done and done well, is found in the lesson on preparing microscope slides. That's the earlier lesson, which you can check. Now, so epidermal cells are prepared, ready, and here. So this is how the cells, epidermal cells here, were gotten. These cells are cells that were placed in isotonic or hypotonic solution. These are the strips that we looked at under the microscope, and we saw this. The cell is colored, the vacuole is colored, and gives the whole cell colored. These epidermal cells, this is the stomach and gut cells. We'll talk about them another time, but that's the epidermal cells in isotonic or hypotonic solution, turgid cells. These are epidermal cells in hypertonic solution, plasmolysed. See, plasmolysed cells. Well, we are now going to discuss the process that led to these turgid or plasmolysed cells. That's the osmosis process. So we have here this illustration that we have made. This is a plant cell in a solution on a microscope slide. So this plant cell is placed in a hypotonic solution. Now, the hypotonic solution, we have explained the key here. The hypotonic solution has a high water potential, this one, and the cell contents has a lower water potential because the hypotonic solution have fewer solids. Here, higher solids. So water molecules moves from the region of high water potential into the cell, the region of low water potential across the partially permeable membranes, and that is osmosis. And notice that when the water molecules move, they go to fill the vacuole. And so the vacuole will keep on expanding, becoming bigger and bigger, and the cell wall is slightly elastic, expands to a point that the cell wall cannot expand anymore. The cell is filled up and is said to be turgid, is firm, is hard. The vacuole is big. Now, this is the situation that produced these turgid cells. Well, we come back on the other hand to look at the cell placed in a hypertonic solution here on the slide. So water molecules move from the region of high water potential, which is the cell contents, into the hypertonic solution. Water molecules move from the vacuole, and water molecules keeps on moving out into the hypertonic solution, across the partially permeable membrane, until the cell becomes soft or flaccid or flabby. Then if the cell continues to stay in the hypertonic solution, water molecules keep on moving from the vacuole shrinks and shrinks, pulling the cytoplasm away from the cell walls. And we say the cell is plasmolysed. And this is the situation that produced these cells. These cells are plasmolysed. And these cells, if are taken from the hypertonic solution and put into water quickly, the water molecules will move in and the cells will regain their turgidity. So plasmolysis can be reversed early if the cells are removed from the hypertonic solution and placed into water. Good. So we have two quick questions to solve. Here we have two cells, cell A and cell B, which are photomicrographs of rhodoscalar cells. Photomicrographs, we discuss them under microscopes. A photomicrograph is a photo taken from a tissue under optical microscopes. So this cell and that cell are photomicroscopes, and we are supposed to suggest titles for A and B, provide titles, and the question 2 is to label all these, provide labels for all these. Okay? Right, so let's go for the solution. The title of A is a photomicrograph of rhodoscalar cell. We don't know whether this is turgid or flaccid. It's a cell. You see the cytoplasm and the vacuole, they fill the whole cell. Okay, so photomicrograph of rhodoscalar cell. This one is a photomicrograph of a plasmolysed rhodoscalar cell. We see a plasmolysed cell, and that is what it is. Now, what about the labels? Looking at it very well, here is a cell wall. This is a cell membrane. This is cytoplasm. And here is a vacuole or cell sap. The cell sap is the contents of a vacuole. Now we come here, looking at this photo very well. Here is a cell wall. Here is also a cell wall, because these cell contents have shifted away from the cell wall. So here is a cell wall, okay? Then here is a cell membrane, because the cell membrane is still attached onto the cell wall. So here is a cell membrane, but here is a cell wall. What about here? It's a cell membrane, because here is a cell wall. The cell membrane has been pulled away from the cell wall because of plasmolysis. So here is a cell membrane. And what about here? What is it? It's a space. This space is filled with the surrounding hypertonic solution, because when the cytoplasm is pulled from the cell wall here is permeable, really permeable. So the contents of the solution in which the cell is lying, the solution will move in to fill the space. So this is a space. It's not just a vacuum, but a space filled with the solution, hypertonic solution. And this area is the cytoplasm and the cell sap, which we can't really work out where the cytoplasm is or where the cell sap is. So very neat and interesting, isn't it? Well, this is the end of our lesson for today. Thank you very much for being along. Please subscribe. Please give us a thumb if you like the lesson. Thank you very much, and goodbye.